Here we're going to discuss about HIV, which is Human Immunodeficiency Virus. For HIV, it is grouped under the family of Retroviridae. So for this type of virus, it contains a type of special enzyme called reverse transcriptase enzyme. This enzyme is able to convert the viral RNA into viral DNA. For HIV, it is also known as lentivirus. In fact, HIV has two main serotypes, HIV-1 and HIV-2. For HIV, if you look at the structure, it consists of three main structural proteins, which is reverse transcriptase, integrase, and also viral protein R, while the other non-structural protein which will not be discussed in this lecture. First of all, human immunodeficiency virus. From the term you already can know, this virus causes the host lacking of immunity. That's why called immunodeficiency. So this virus only target on those host cell that having CD4 receptor. So what are the cell that having CD4 receptor, which is a T lymphocyte, dendritic cell, and also macrophages. So this virus will only attack T lymphocyte, dendritic cell, and macrophage, as they have the CD4 receptor. However, for this viral to enter into these three types of CD4 cell, it will also interact with the other chemokine receptor, which is also known as the co-receptor. So the co-receptor is known as CXCR4 or CCR5, depending on either it is found in the T cell or either it is found in the macrophage. However, all those cells that having the CXCR4 and CCR5 with co-receptor, they are grouped under CD4 receptor cell. So if you look at the structure, this HIV is contain a type of special enzyme called reverse transcriptase enzyme. This reverse transcriptase enzyme is able to convert the single-stranded viral RNA into double-stranded viral DNA, and then later on this viral DNA will integrate with the host genome to form as a provirus. This will be carried out by integrase enzyme, which found in the HIV. So in terms of pathology, first of all, the patient get contact with this HIV viral via blood transmission, blood transfusion, via sexual intercourse, or in some cases like the needle drug sharing, and also it can be transferred to, from the maternal via the placenta. So from here, the patient will get this HIV virus. So once they get HIV virus already, this virus will interact with the host CD4 receptor. Once it interact with the CD4 receptor, later on, you interact with the co-receptor. So the interaction is via this GP120 glycoprotein found on the HIV virus surface. So in terms of infection, first of all, the HIV will use the GP120 glycoprotein to interact with the CD4 receptor. Later on, will interact with the co-receptor. Once it interact already, endocytosis will occur. So the virus will release the reverse transcriptase enzyme together with the viral single-stranded RNA into the host cytoplasm. Once a viral reverse transcriptase enzyme and also the viral RNA is released into the cytoplasm, this reverse transcriptase enzyme will convert the single-stranded viral RNA into viral DNA, double-stranded. Then later on, what is happening is this viral DNA will move into the nucleus of the host genome and fuse with the host genome, which means it will fuse with the DNA of the host cell, the CD4 cell. How this can be done? Actually, the viral DNA need to pass through this nuclear pore found in the nuclear envelope. So for them to do this integration, you need a pre-integration complex. One of them, the most important, is integrase enzyme. This integrase enzyme allows the viral DNA to fuse together with the DNA of the CD4 cell. So from here, a copy of the proviral DNA is integrated into the cellular genome at the random site. So from here, the virus is forming provirus. So at this point, this HIV virus will remain silent in the patient for many years. So that's why for those patients with this HIV infection, actually they can survive for many years, because during this period the virus remains as a pro-virus, which means this virus is just like sleeping, which is in inactivation stage, in inactive stage. So during this inactive stage, it will not going to destroy or cause any major changes in the immune system of the whole cell. So that's why the patient can survive for many years. For HIV infection, the clinical stage can be classified into three. So first of all, we have a primary acute infection, we group that as A, which means the person is developing mild acute influenza-like illness, such as this flu. And then the patient may have developed this viremia. Later on, the virus will spread throughout the body. So there's a vigorous immune response developed, reducing the viral load to set point, which means it will make the virus number decrease to very minimal amount. While some of the patients later on they will develop into this chronic symptomatic, which group as B. So this normally happen in those asymptomatic cases. So they are moved to the stage B within 2 to 4 years. So for untreated patient, they will progress to clinical stage C. So in the clinical stage C, the patient is showing the advanced AIDS, which means the immune system is already been destroyed by the HIV, and then the patient normally will die due to other type infection. So in fact, for those patients infected with this HIV, they die is not because of HIV virus, but it's due to the weakened immune system, and then die because of just like normal flu or cough. So this picture showing the clinical and virological event during the HIV infection. So those patients is showing the symptom developed in those patients with this primary acute infection, chronic asymptomatic, chronic symptomatic, and also advanced HIV, advanced AIDS. So normally, for those patients with this HIV infection, okay, the HIV will destroy the immune system of the host. So by this, just a normal flu will cause death for this AIDS patient. However, according to the statistics shows that quite a number of AIDS patients they die because of this tuberculosis. So in the stage C, the clinical AIDS patient, they are exposed to the opportunity, example like those of the malignant diseases and also the infective manifestation. So this patient will have suffering from all of these type of infection and some of the malignant disease. So in terms of immune response, so the patient will develop anti-GP120 antibody. So this antibody will develop slowly and it remain high throughout the infection. So that's why in the laboratory diagnosis, most of the time we want to know whether the patient is having HIV infection or not. We will look for this antibody against the GP120. While the other antibody the antibody against GP24, actually this is against this internal capsid protein. However, most of the time we only look at the presence of GP120 antibody. Well, if you look at this one, what you will find that in acute infection, once the patient is infected with HIV, the viral number increase dramatically, and then the CD4 cell number will drop. However, you may not be noticed that later on the patient will start to develop antibody against HIV. So this increased amount of antibody against HIV successfully bring down the number of HIV virus in the patient. So it looks like the host immune system is already win in this battle. In fact, it is not. Why? If you look at this curve, the number of these viral particles is very little in the body, in the blood. Actually this is because the viral form provirus in the nucleus of CD4 cell. So this makes the host immune system is unable to trace the presence of this HIV. So it looks like the immune system is already win in this battle. In fact, it is not. Wait until several years when the immune system, when the provirus is get activated. So this large number of HIV virus will coming out from the CD4 cell. At this point, it will rupture the T lymphocyte. Once the T lymphocyte rupture, you will be noticed that there is reduction in the number of this CD4 T cell. So when there is losing of this CD4 T cell, the host will be losing cell-mediated immune response and humoral immune response. So that's why at this point, the patient will develop this compromised stage, and then lacking of immunity will cause the patient die because of other infection. So this is in terms of the epidemiology. So in fact, first of all, it was suspected the virus is spread from the chimpanzee. And this is a global situation and the trend of this HIV. So for laboratory diagnosis, so we can diagnose this HIV infection using ELISA. While the other gold standard to diagnose HIV is use RT-PCR, because this virus is having an this RNA as genetic material. So by this, using RT-PCR to diagnose HIV is a gold standard. However, for laboratory, ELISA itself is also sufficient enough to diagnose HIV. So for in terms of treatment, there are several treatment. However, all these treatment and the potential treatment, they are just in testing and clinical trial. There are more this type of this therapy are in research so that they are looking for a better chance for this HIV treatment.